Welcome. I'm pleased to be introducing the first speaker of the Fall 2023 Great Explorations Seminar. And the theme for this series is sustainability. Our first speaker is my dear colleague and friend, Professor Matthew Wells, who's a professor in the Department of Physical and Environmental Science at the University of Toronto. Professor Wells obtained his PhD from the Australian National University in 2001, where he used laboratory experiments to explore fundamental geophysical fluid dynamical processes. His research was complemented by fieldwork with two oceanographic voyages to the Antarctic and to the tropics. After completing his PhD thesis, he had the opportunity to work as a postdoctoral fellow at the Technical University of Eindhoven in the Netherlands, and then in the Department of Earth Science at Yale University. He joined the Department of Physical and Environmental Science at the University of Toronto as an assistant professor in May of 2006, followed by tenure and promotion to associate professor in 2011, and then to full professor in July 1st, 2021. Professor Wells has nearly 80 publications in top-tier journals in limnology and geophysics, or more specialized journals in fluid mechanics. In addition, he has an impressive funding support from a number of provincial, national, and international granting agencies that has enabled him to establish a world-class, and I cannot stress that enough, research group at UTSC. He was the recipient of the highly coveted Early Researcher Award from the Ontario Ministry of Innovation and two Answer Discovery Accelerator supplements in 2011 and 2016. Those are very competitive, and I think this speaks volumes to the transformative science that is being produced in his lab. He's also more recently the co-director of the Water Pathways, an excellent initiative associated with the UTSC research clusters of scholarly prominence, which water-related research.
But if they want the link,
Someone disagrees with what I'm saying? Okay, today Matthew will present on lakes in a changing climate. This talk will offer an introduction to the key physical processes that structure lakes in different seasons. And we will discuss how fish stocks and water quality in our lakes could change in a warming climate. Trust me, you're all for a treat. Without further ado, the stage is yours.
Thank you, George. Is the microphone on? This mic? Okay. So George didn't mention this. We actually share a birthday together, and we got hired at the same time. So and we live near each other. So it's been George has been a real pleasure to be in this department for the last 15 years. So today I'm going to give a introductory talk about some aspects of lakes, and I will talk about how the physical dynamics of lakes influences some of the distribution of fish and biology and plankton in the lake. And so you might be wondering, what's this tank I've got up here? So I used to do laboratory experiments, and they're kind of fun, but I still use them mainly for teaching, but I do a lot of field work now. So at the end of today's talk, what I want you to understand is a picture like this about how the thermal structure of the lake influences the mixing. And about halfway through this lecture, I'll do some demonstrations here to show how small density differences within the tank can actually inhibit mixing throughout the dynamic layer. So at the back, are you able to see this, this tank? I tried to raise up the table, but I'm not sure if at the back you can easily see it. Okay. So the topic of this seminar series last semester and this semester is on sustainability. Is that right, George? So I thought I'd put up what the Sustainable Development Goals of the United Nations are. These are partly motivating the Institute for Resilient and Inclusive Societies and Ecosystems at University of Toronto. We have 3 institutes now, and they're motivated by the many Sustainable Development Goals the UN has come up with, which we're aiming as a society to reach by 2030. Now, you notice a lot of them are on very different topics. Last semester, I think many of the talks were on more the social sciences. The talks this semester are more on the physical sciences. So there's 2 in particular of these 17 worthy goals that I'm going to touch upon, and that's namely clean water and sanitation and life below water. So, this is an effort that hundreds of countries around the world are working on. It's not something I'm doing by myself, but at University of Toronto Scarborough, we have this group called the Water Pathways Research Cluster, which is aiming to address some of these issues. So in particular, these are the 10 professors that are involved with the Water Pathways Research Cluster. You'll notice George Arenditz is here. Next week, I believe my colleague Cosima Porteous is giving a talk about the changing chemistry in water bodies and how that's affecting fish's ability to smell and reproduce. And then later in the semester, Professor Carl Mitchell will talk about, he'll talk about methyl mercury, how that goes into the water and how that is a problem for water quality. And these other researchers are doing really exciting work as well. So I just sort of put that up there. Okay, so where is fresh water around the world? Some of it's in rivers, some of it's in the groundwater, and some of it's in lakes and reservoirs. So I made this word cloud, took all the papers I'd ever written and took words out of the title, and I was intrigued to see that lake is actually the most common word in any of my books. So I'm going to talk about lakes. I'm not going to talk about groundwater or rivers. You might see some of the other words. A lot of these words are kind of physical words like stratification, circulation, Coriolis effects, density. So my work is sort of motivated by how physical processes control the movements of water and then how that structures where the fish are, where the oxygen is, what the water chemistry is. So I have a background in mathematics and physics, and mathematics and physics is fun, but it's a lot more fun going out and engaging with community stakeholders and working on boats. So so this is sort of the general focus of my research today. So I'm originally from Australia, and if you've ever been to Australia, the thing you'll notice about Australia is it really lacks a lot of water. You don't have to go far from the coast to a lot of it's desert. And when I was doing my PhD in Australia, that whole decade of the 90s, it was really a long-term drought. There was severe water restrictions, great lack of water. So we were very conscious about drinking water, having showers, washing our cars. And then I moved to Canada 15 years ago. And Canada's a lovely country, but one thing that's really, if you've grown up in a country without water, one of the things that's really impressive about Canada is how much water we have. Most of it's really good quality, and we've probably got, so this map is the density of lakes around the world. So a lot of the Canadian landscape is dominated by lakes. It's really, I guess foreigners often think of Canada, the Rocky Mountains or all that, but the thing that really defines Canada as a country is we have so many lakes, so many freshwater resources, and we're really blessed by them. So if you look globally at the whole world, there's really sort of 4 lake systems: Lake Baikal, the African Great Lakes, and Laurentian Great Lakes, and the Caspian Sea is technically a lake, although it's salty. These bodies of water hold most of the world's freshwater supply. So Canada's got not both the largest lakes in the world and also a landscape that's dominated by lakes. So I like being in Canada because it's a wonderful place to study lakes. So what are the things we worry about? So many of the lakes in Canada are beautiful and pristine, but where there are population centers and where there's agriculture, there's the potential for sources of pollution. So the sources of pollution could come from cities. We have wastewater treatment plants that we like to pretend don't exist, but they're an important part of our lives. And these discharge into the rivers. We have factories. There's animal feedlot. We all like to go to the supermarket and have abundant fresh food, milk, eggs, beef. That has a source of pollution: cropland, forestry changes, and then road salts runoff. And then also we take water from the lake to drink. And so if you don't have to look very hard, down at the end of Highland Creek, if you go the very end, there's the Highland Creek water treatment plant. If you go a few kilometers west, there's the major water take in that brings water out of Lake Ontario for us to drink. So how do we do this? How do we both use the water as a resource that we're drinking and as a place where we put our waste into it? So it's important to distinguish what are point sources. So the water treatment plant is a point source. There's a particular pipe that comes out. There's someone who's responsible for this, and this is regulated. for the last 50 years. Factories are regulated as well, but on-point sources are not regulated. So the fertilizer that goes on crops can wash into rivers and end up in the lakes. Runoff from roads ends up in the lakes. And I understand from conversations with George that animal feedlots, while they look like point sources, are actually not treated as point sources. They're not regulated. So there's a lot of things we do regulate and don't regulate. This all washes into the lake. It affects the fish habitat, and it also affects us directly because we drink the water. So how do we do that in Canada to understand it? So this is really this question of, it's what motivates a lot of us in water pathways. What is the processes in the lake? How do we manage these to have good water quality for fish and for drinking water in the lake? So the point sources have been regulated, I think, since the 70s through the Great Lakes Water Quality Agreement that was signed between US President Nixon and the Canadian Prime Minister Pierre Elliott Trudeau. So they've been largely fixed. You know, the water we discharge from our wastewater treatment plants meets standards. What's not regulated are agricultural. So there is a relationship between the economy, how productive the economy is, how ability to get cheap, reliable food, but also the downside is that how do we do that with avoiding poor water quality? So in the Great Lakes, most of the Great Lakes now is pretty good compared to what it was in the 60s, but there are regions that are not good. And particularly, so we're here in Toronto. If you go to Lake Erie, a lot of Lake Erie suffers from algal blooms, and these appear to have been increasing over the last few years. So why have we spent all this money since the last 50 years? We've fixed up all the point sources. Why are there algal blooms occurring? And so it's actually some of the richest agriculture in the world is in Ohio and Southern Ontario. That's the other thing I like about in Canada. You know, I can go for a drive and you see fields of beautiful fields of wheat and corn, as opposed to Australia where there's grass every 2 feet. So this is a hugely important economic region where we grow food, but the non-point sources drain into the Thames River, which flows into Lake St. Clair. And you'll notice on the American side, the water coming down from Lake Huron is nice and blue, but on the Canadian side, we get these algal blooms. So often people, at the moment the summer starts, the area gets warm, all the beaches are closed, you're not allowed to swim in it. Down here, we have the Maumee River that comes in from Ohio. It's a large area of production of corn. Corn goes to feed cattle and other livestock, and this drains into the river and causes really severe algal blooms. So in Toronto, it's easy to forget this. If you haven't been, I'd really encourage you to go and look at the Harrison water treatment plant at the eastern end of the Toronto beaches. It is the oldest, and it supplies about a quarter of the water in Toronto. So they draw water from about 20 meters, about a few kilometers offshore, 20 meters down. It goes through this plant, through a series of filtering and sterilizing things. But when I've done tours with my students, the guys said the water's pretty good in Lake Ontario. We really don't need to do much to make it drinkable. Like, they screen out it and they filter it and test it all the time. But generally, most of the time, the water in Lake Ontario near Toronto now is pretty good. So it's easy to forget how privileged we are. But if you go, and so part of the reason it's good is the water is really clear. So here, we're on a boat with some students. We lowered a GoPro camera into the lake. And the point I want you to look at is just how clear the water is. We can look down from 20 meters up to the boat. I know it jiggles around a lot, but the point is you can see the boat. We're looking up from deep within the water, and the water is so clear, it's easy to see. So there's very little nutrients in Lake Ontario, very little algal mass, and no problems with water quality. I tried to do this a few weeks ago with my students, and I didn't tie the GoPro camera on properly, and now it's sitting on the bottom of the lake. So apart from that, it's pretty easy to do this. So in Western Lake Erie, though, the water quality does not look like that at all. If you are brave enough or foolish enough to go swimming, you might be able to see 1 or 2 feet in the water. So here's an aerial photograph near Toledo of an algal bloom. The water often looks like your lawn. It's a shallow lake. There's abundant sunlight through the whole water column. There's a supply of nutrients in, and the water looks looks poor color. You wouldn't want to go swimming. But sometimes recently there's been these incidents that you probably heard of 10 years ago where Toledo had to shut off the water supply for several days. They had to say you cannot safely drink the water. It contains this toxin called microcystin, which I don't think any people have died of it, but cattle, when they've drunk it, have died from it. So they're very cautious, and even boiling the water will not treat it. So for several days, the city of 500,000 people did not have drinkable water, which in a country like Canada, we like to think, well, in a country like America or Canada, we like to think shouldn't happen. So broadly, a lot of us who study aquatic water systems, George Arenditsis, myself, and many other hundreds of scientists around the world, we're trying to understand what's the difference between these systems that do form large algal blooms and Lake Ontario that has very good water quality. How do we change regulations so the nutrients don't result in these algal blooms? And so more broadly, while big cities in Canada generally have pretty good water supplies, there are a lot of communities in Canada that don't have good water supplies. A lot of the smaller First Nations communities across Canada and other small communities have these boil water advisories. And so it's a bit disappointing, I guess, that, you know, as a rich first world country, we can't provide good, reliable drinking water to every citizen in this country. So that's kind of motivating why this is an important topic to do. You know, in Toronto, we can just take it for granted. We turn the tap on, you can drink all the water. You know, most countries in the world, you can't do that. Even a lot of communities in Canada, you can't do that. So this is a difficult problem. I think it'll keep George and I busy until we retire, and the next generation too. But the question is, if we know what these sources are, how do we understand how they affect both the fish habitat, where algal blooms, and what the oxygen levels that fish need to breathe and respire in the water are? So the rest of the talk's really going to be how the mixing dynamics in this layer influence where you might get algal blooms and how that might shift in climate change. Okay, so what are the physical process that I'm really most interested in is how water often doesn't mix. So often when you look at a tank of water like that, your sort of default assumption about water is that water mixes, you know, water homogenizes, and the sort of naive expectation is if we have nutrient sources going into the lake, they will just dilute and spread out uniformly through the lake. I think most of you, that's probably how you think about water. You know, you've got your coffee in front of it, you mix some milk into it, it homogenizes. But one of the defining features of natural water systems in the ocean and lakes, even the atmosphere, is that often water or gases do not mix very well. And so the reason they don't mix is because they can actually form layers. And so the demonstration is going to show you that how we form layers in a tank. So in the ocean, what I used to study is layering occurs because you have fresh water and salt water and warm water and cold water. Lakes are a little bit simpler because most of the time we don't need to compare out the salts. There are salts, but they're very small and they don't affect the density. So this exciting graph is one of the ones I think about a lot. On this axis, I have the temperature. On this axis, I have the density of water. So water, is that still working? Yeah, so water is actually what the metric system is defined upon. So 0 degrees in Celsius is where water freezes, 100 degrees is where water boils, and the maximum density of water is defined to be 1,000 kilos per cubic meter. So that's not chance, that's the French in their infinite wisdom define the system around water. So, you know, Australia went metric in 68 before I was born, so I've always, you know, I've only been exposed to Fahrenheit when I moved to the United States. Fahrenheit doesn't make any sense for water, but for density it does. So the maximum density is 1,000, and then as you warm the water up, it gets lighter. And so you have lighter water, heavier water. These are actually pretty large differences in density. So by being a few kilos per cubic meter lighter, it can float on top of the other layer. Now you might notice there's also a small decrease. It's maximum density at 4 degrees, and then it gets slightly lighter as you go to 0 degrees. And that turns out to be really important in Canada. In Australia, we didn't need to worry about this because it never gets below 4 degrees. But in Canada, we have frozen lakes. This part of the graph turns out to be really important for what happens for many months of the year. So with that in mind, I'll show you this little demonstration I dragged up from my laboratory. So we've got a tank of water at the moment. But what we can do is we can add some, rather than change the temperature, I'm going to add some salt and add some alcohol. The salt will make the water heavier and it'll sink. The alcohol will make the water lighter and spread out at the top. So first I will add some, this is all food dye that you put in cakes if you're making them for kids. Make all those horrible colors for Halloween. So I'm just going to add a small amount of salt. This is only 50 grams roughly I'm adding. So this is how you normally think of water. You mix it, it homogenizes. So here I've added a very small amount of salt. It doesn't change the density much. But what we'll see is that this will sink to the bottom of the tank and spread along the tank. It'll be a bit of mixing, but it'll stop and it'll form a layer. I'll pull this out. And it will slowly take about 20 seconds to spread along the bottom of the tank. So I spent years studying this when I did my PhD. I got a bit bored with it, but it's still fun to use for teaching demonstrations. Okay, so it's spreading across the tank. You can describe how much mixing occurs, how fast it goes. It'll come to the other end of the tank, it'll sort of rise up, and then it will oscillate down, and then after a while it will stop, and it will stay like that. Molecular diffusion is really slow, and so we could come back the next day, and this would still be sitting on the bottom of the tank. So it's rising up, and then it will sink down, and then we'll have a layer that will persist for the next hour or so. So I'll wait till that comes back, and then I'll put some more dye. Okay, so now I'll try and put about twice as much or 3 times as much salt in. And I'll make this one blue. Okay. So this is actually heavy. It'll go underneath that green layer. There'll be a bit of mixing, but the point is the green layer will persist. We'll end up with a situation where we've got 2 layers. So you see it's denser, it's moving a lot faster now. You see how the top isn't moving at all, but we're getting these big internal waves going up and down within the tank. So there has been a bit of mixing between these 2 layers, but there's still a distinct green layer and there's still a distinct blue layer. So now what we'll do is we'll add a something light to the fluid. So this is not cold water we added, it's like adding cold water. Now I'll add some alcohol, which is like adding warm water. So I'll pour this in at the top. I'll dye this one yellow. Just going to mix this a little bit. Okay, so this is light, it'll spread out along the top. Slowly. And then I'll put a red layer in too. There's a reason I'm doing this, because the next few pictures I show have all got a little rainbow in them. So I just want you to like have this rainbow to look at, to think about. Okay. So what I'm hoping you see here is we've got about 5 distinct layers. We've got this layer at the top, a yellow layer, an intermediate layer, and they're organized by density stratification. You'll notice some like waves going up and down, but this is pretty stable, and it'll sit here with various layers. So the word we use for this is density stratification. So if you imagine I was on my little boat and I lowered a probe into the water, I could make a graph where I had, let's say I had the temperature. We'll pretend these colors are representing the temperature. And then this axis is the depth. So what we'd find is that the top layer, it was relatively, warm. And then there's another layer, and then another layer, and another layer. So if you imagine these had chemicals in them, only the top layer is in contact with the atmosphere. So if you want to know about oxygen, oxygen can get into this top layer, but it's inhibited of mixing down to these deep layers. And if we had biological material decaying in the bottom layers, it could suck out all the oxygen in the bottom layer, and there'd be the barrier to mixing between these layers. Similarly, if you had plankton growing in the top layer, they could stay mixed where the sunlight is, but not be mixed down to the bottom layer. So this density stratification turns out to really limit the physical transport between layers, which then has a direct impact on the chemistry and the biology. So when I go out on a boat, often I measure the temperature with fancy probes, or I have loggers that are in the lake measuring the temperature. Then I work out what the density is, and then I'm really interested in what the structure of the density layer within the lake is. So the reason I went to a lot of trouble to make this picture is the thermal structure changes during the year. So this will persist for a while, but now I've got at different times of the year, you have different thermal layering within the lake. This has different mixing dynamics, and this has a direct impact on what the oxygen is in the lake, where the plankton can grow, and this then influences where the fish are in the lake. So if you want to understand the context within which water quality changes are occurring, the physics is sort of fundamental to describing where water quality issues can occur. So I've got a series of graphs showing what happens at mid-latitudes where we are now, what happens up higher in the Arctic, and what happens further south. So this is a picture I drew where we're looking at summer, fall, winter, and spring. And like this picture here, that the colors are representing the temperature. So we've got warm water, medium water, cool water, and then very cold water near Fearing. So it's freezing. So it's sort of a bit like this picture here. Okay, so during summer, when most people, I should have asked, how many people go fishing or boating or swimming in the lakes? A lot of you? Many of you. Okay, so during summer, it's quite nice to go swimming in Lake Ontario. The water can get up to 23 degrees. Most of the lakes in southern Ontario, the very surface is really warm. But you might have noticed if you've been at one of a small lake in the cottage, you don't have to go down very far, like even 3 or 4 meters. If you dive down, you can feel the water getting colder. So what's actually surprising in many of the lakes is actually only a thin skin in the top 5 or 10 meters, it's warm. And so this is the layer where the sunlight penetrates down. And then if you go down, here I've got a diagram where I've got temperature on this axis, depth on this axis. The temperature drops very quickly. And so the bottom of most lakes is actually really cold. It's very close to 4 degrees. It's very close to that maximum density. There's dense water that sits on the bottom of the lake. So right now in Lake Ontario, a few weeks ago, it was still nice to go swimming. Right now, the air's getting colder, the surface layer is getting deeper, and the wind's driving it mixing down. So right now, we're in this period where the water column's homogenizing, and it's going down to the bottom of the lake, and soon it'll go down below that 4-degree level. And so in winter in Lake Ontario, in January and February, we actually get what's called an inverse stratification. The coldest water is now at the top. So below 4 degrees, it actually gets lighter again. So in Lake Ontario, we get a layer that's about 20 meters deep, where it goes from 0 to 4 degrees, and the whole rest of the lake's 4 degrees deep. So as you go from summer to winter, there's one period of mixing. In the springtime, as we warm the lake up, we go above this 4-degree period. And so there's a brief period of mixing when we go below 4 degrees to 5 degrees, when the wind can overturn the whole water column, and then we stratify again. So these lakes are called diamictic lakes. There's twice a year they mix. They have a very prolonged period in the autumn where the whole water column homogenizes, and then a short period in the spring. So what climate change could do to this, it could change the length of the summer stratification, and it could potentially reduce the amount of time you have to mix in the fall. So some of the work we've been doing in the Algonquin Park, there's actually been a 6-week shift in when the fall overturn occurs. So if you go back to 2000, this fall overturn would occur now in late September, October. For the last few years, it hasn't been occurring until October, November. So this duration of the physical mixing is shifting with climate change. In winter, what we're seeing is the length of ice durations changing, and it's also changing the duration of how So with climate change, the amount of time you spend in each of these quadrants is shifting. This is getting longer, this is getting later, this one's getting shorter. If it got really warm, you could flip to a system where rather than mixing twice a year, it might only mix once a year. So if you go a bit further south from Ontario into New York or Pennsylvania or continental Europe, where the climate's a bit lighter, it might not get very cold in winter. You might never get down to freezing, and so you might never actually cross this 4-degree line. So these lakes, which is typical of lakes in from about 40 degrees down to 20 degrees, during summer they're stratified, and then one mixing period in winter, and then they stratify again. So these are called monomictic lakes. So what might happen in Ontario in 20 years' time in lakes that currently mix twice a year and have a winter stratification might transition to these lakes that only mix once a year in winter. So that's what a lot of lakes further south look like. If we go further north, so really far north up to the Arctic, to somewhere like Great Bear Lake in the Northwest Territory, they're really cold most of the time. And so in summer, they barely get above 4 degrees. In the fall, they cool down again. They stratify in winter, and then they mix again in summer. So this is also a monomictic lake, but rather mixing in winter, now it's mixing in summer. So we had a workshop last week, and a colleague from British Columbia came and gave a talk about Great Bear Lake. Great Bear Lake said this transition, it might change from being a cold monomictic lake where it mixes once a year to start looking more like the lakes in Ontario. There's also lakes that mix all the time. So the western lake of Lake Erie is very shallow. It's only about 8 meters deep. It's not much deeper than this room. And so every time the wind blows, you can mix the lake. and then the sun comes out and it stops being windy and it can actually get stratified. So one of my PhD students is looking at this, and basically every week it oscillates between being stratified, mixed. It's so shallow, it doesn't take much for the wind to mix it. So we call these lakes polymictic lakes. So if you know these four words, polymictic, dimictic, monomictic, or monomictic, that describes most of the lake dynamics in the world. So what I'm really trying to do is think about what actually happens with this mixing dynamics here and the stratification dynamics there. Any questions about any of that? Yep.
Talk about layers, but it seems to me it's a constant change.
Yeah.
Where do you draw the line on some to figure out what the different layers are?
Yeah, that's a good point. It's a bit arbitrary. So when you go out and you sort of idealize these lakes as we're having a temperature profile that looks like that, that, and that. And we have these three distinct layers, an epilimnion, a metalimnion, and a hyperlimnion. And so conceptually, that's how we think of it. But in the real world, nothing has these distinct categories. So, usually the profiles kind of look a bit like, they're sort of continuous. So often we're interested where the maximum temperature gradient is. And I wrote a whole boring paper about how you actually go to the definitions of it. It does matter if you're trying to compare two lakes that have different geographical locations, or you're trying to compare one year with the next, you have to have consistent definitions, but it is a bit problematic to say exactly where these layers exist. Yeah, so that is, but conceptually we like to give things distinct labels, but it's all water, it all bleeds into each other, so it's not, in this tank here we had 4 distinct inputs, but there was mixing between them, and so if you actually measured it, it would have a continuous transition between these layers. You got a question, George?
George is noticing the question online. Thank you, George.
Oh, okay.
So from someone who's watching us over Zoom, where do meromictic lakes fit in? Are they lakes with no mixing?
Okay. There's endless categories of lakes. There's a type of lake that's been in the press a little bit lately called a meromictic lake. This is a lake which has some chemical stratification. So in Ontario, there's only a few places where this naturally occurs, but sometimes this can occur. You might have been to Grenadier Pond in High Park. That's right by the highway. It gets a lot of road salt going into it, and it actually becomes chemically stratified. So the bottom layer is not dense just because of temperature. It's dense because it has extra salt in it. So meromictic lakes have been in the news a bit lately because there's a lake called Crawford Lake on the Niagara Escarpment. It's meromictic, it doesn't mix. It turns out this is really important for preserving the sediment record. And so it was after a decade of research, it was chosen as the golden spike for defining where the Anthropocene is. So this was a big deal this summer. This lake on the Niagara Escarpment is like the lake that defines the modern era of anthropogenic change in Ontario. So the word meromictic has been in the press a little bit lately. That's possibly where the question's coming from. So meromictic lakes are pretty rare. They don't occur very often. And so that's why I haven't introduced them in this little discussion. Okay, so why it's important to know if they're a dimictic lake, a monomictic lake, a polymictic lake, or a oligomictic lake that might mix every few years is this is really where the lake breathes. So you tend to think of lakes, well, you guys don't tend to think of lakes at all, but you often think of lakes being mixed by the wind every time. But if the wind was blowing, and I'll show you this later with my hairdryer, I would only mix the surface layer. So you only mix the whole water column in a dimictic lake twice a year. So that's like the whole lake is breathing in fresh oxygen. And so if we have a monomictic lake, it has quite a different oxygen environment. And so the fish that do well in those lakes have to tolerate lower oxygen levels. So the fish that do well in Australia are adapted to lower oxygen levels. Canadian lakes, typically they're deep, cold, oxygenated water. So the fish like cisco and trout that are prized by sports fishing, they've evolved to require oxygen. If they don't have oxygen, they don't do well. So if we had a shift from dimictic lakes to monomictic lakes, the amount of oxygen that they're breathing in would be very, very different. And then these warmer lakes, like polymictic lakes, they could potentially breathe all the time. So that's a nice way to think about why the mixing regime is really important, because it's sort of how the lake's breathing, if you like. So the simplest effect of climate change is we're going to change how lakes breathe. Lakes that are currently dimictic might warm up and become monomictic. Rather than mixing twice a year and having lots of oxygen in the deep waters and cold waters, bottom waters would be warmer, and they would not have as much oxygen. So there's some suggestions this might be changing right now in Lake Michigan. There's been a few years in the long-term records of Lake Michigan, the whole lake hasn't completely homogenized. We might be in the next 20 years sort of shifting a monomictic system. Then if you go really far north, speaker last week was talking about Great Bear Lake, which is at least as big as Lake Ontario. It's a lot deeper. It's a massive lake in the north. At the moment, it doesn't get above 4 degrees. Next 20 years, it might transition to being a dimictic lake. So it'd have quite a different oxothermic regime. So these are, and like you said, I mean, I've given these names categories, but of course there's a continuous change between them. So these shifts will have a profound effects on the oxygen regime, the plankton, and the fish habitat. So just as an example, you can't easily go out in a boat and measure this yourself, although I did see a colleague on Twitter recently was saying how her Apple Watch measures pressure and temperature, and she loaded on a string in Lake Washington and Seattle. She could measure temperature profiles, but the American National Oceanographic and Atmos Agency, they have an office in Ann Arbor, Michigan, where the GLERL, the Great Lakes Environmental Research Lab is, and they put out live forecasts of water currents, water temperatures of the Great Lakes. So this is from a week or two ago. We've got a picture of Lake Ontario here, and they're showing slices through the lake at different locations. And so what you see here are the colors: cold water at the bottom, warm water at the top. If you look at it today, I suspect this warm layer is mixed down deeper. In the next few weeks, this whole water column will homogenize and mix. So at the moment, Lake Ontario is going through the overturn season in fall. Usually no one goes out.
Yes.
Usually no one goes out in winter, but in winter, measurements we've made suggest there's an inverse stratification. It gets close to freezing at the top, and then the bottom's also 4 degrees. So there's this very large layer in the lake. Now, one thing you have experienced, so most of you haven't measured this. Maybe go fishing, you might have seen it, but you have drunk the tap water from Toronto. And so the tap water from Toronto, one thing that really surprised me when I moved here was how cold the showers are. You know, if you don't put the heating on, it's really cold in the showers. Like, why is it so cold? So it's so cold because the water we're drinking is actually coming from that deep layer. So recently they replaced the pipeline for the water intake on the Toronto Island. And here's a picture of them dragging out the several kilometer pipeline. Then they sunk it and went down to 80 meters. So it's drawing water from 80 meters depth in Lake Ontario, which if you look at the picture, it's this cold water that's sitting deep within the lake. There's a few reasons to do it. Cold water is refreshing. It tends to be lower in plankton as well because it's outside what we call the photic zone. So you're not getting any algal blooms down there. And it's generally very clean and reliable. So when you have a cold shower, part of the reason it's so cold is because it's coming from the bottom of the lake. A project that maybe many of you we're not really celebrated enough is that there's a company called Enwave. They take advantage of this cold water to cool down a lot of the downtown city of Toronto. So the water comes in, it gets treated at the island, and then right near the Rogers baseball stadium, is it called the Rogers Centre now? Just by the CN Tower, and they have this massive underground heat exchanger where they use this water they heat up the water from the lake that goes to city, and then they run this through a lot of the air conditioning units in downtown Toronto to cool down many of the buildings downtown. So this works because we have a dimictic lake and we can take this cold water. So without really knowing about the cold water, you've had some experience of it. Also, where nutrients grow in the lake is determined by the stratification. So in summer, when you have this deep layer called a hypolimnion, the thermocline, and the top layer, this layer up here is in contact with the sunlight. Nutrients tend to be down the bottom, and so they often tend to get a maximum amount of chlorophyll at the bottom of this surface mix layer. And so I've shown, does anyone know what this is? Yeah, so some of you might have a cottage up north and you might have a cottage association. Often to measure the water clarity, we use this device called a Secchi disc. It's named after an Italian astronomer of all people, who was charged with coming up with a simple device to measure water clarity. So what we do is we lower this into a lake and you lower it down until you can no longer distinguish between the white and black quadrants. And so it's a very good measure of both the abundance of plankton in the lake and also where the exponential decay of light goes to about 1% of surface levels. So this is often used as a symbol for limnology. So you need to have both light and nutrients, and so the stratification is really important for where this algal bloom occurs. So if you have a lake that has fairly low levels of nutrient, the Secchi disc often tends to be quite deep and you tend to get fairly small algal levels. And so this is what most lakes in Ontario looked like before widespread agriculture after European colonization. So if I measured the oxygen in these lakes, I'd actually have a maximum oxygen at the bottom of the lake. And that's because the bottom of the lake's cold. Cold water can absorb more gas. And so the highest levels in the lake were at the bottom. It would get up to like 12 or 14 milligrams per liter. And so that's what a lot of fish have evolved for. Trout and cisco and salmon, they want to be in this cold, oxygenated water because that's the habitat that's there all year. Now what's changed is with the introduction of a lot of wastewater treatment plants, septic systems, agriculture, we have a lot more algae produced. And so we have a much bigger, it's a lot greener. When this dies and sinks down to the bottom of the lake, it sucks oxygen out of the water to decompose. And so you have quite different oxygen profiles in systems where there's consumption of oxygen as plankton decreases. Rather than the bottom of the lake having the most oxygen, these systems now have no oxygen in the bottom of the lake. So we have a completely different type of curve. So rather than having maximum at the bottom, we have no oxygen in the bottom of the lake. And so in part, this is driven by the dynamics of how long this layering occurs, and it's also driven by the various point sources and non-point sources of nutrients that determine how much plankton grows. So broadly speaking, I do this part and George does that part to characterize our complementary research programs. So what we don't know much about though is winter. So typically for the last 50 years, academics, they don't teach in the summer, it's nice and warm. We go out on lakes and we measure what happens in summer. But moving to Canada, one of the really defining features is, from about now until April, it's going to be cold. And so this is a time of year when academics are busy teaching, the marina's shut, it's hard to go out and do field work. And so winter hasn't really been studied much. So this picture of what happens in lakes with the oxygen, that's a summer story. Most of the research that we do in North America and Europe is in summer for a multitude of cultural and logistic regions. But something I've been getting interested in recently, what happens in winter? Winter's a big part of the story. And so on this particular day, it was minus 10 degrees. Our gloves were sticking to the boat. It was actually a bit scary to go out and work. If it gets a little bit colder, you can go on skidoos and you have a nice platform to measure the lake. But a big question is, what happens to oxygen dynamics during winter? Is that weak stratification actually matter? It seems really puny, that difference between 0 and 4 degrees, but it turns out to be kind of interesting. So this is another video of a GoPro camera looking up through the lake. And the main thing you notice is the water column's not blue, it's green. And so it's green because there's a lot of algae in it, but it's also quite clear. So as I lowered this GoPro, this is on a temperature logger. I'm slowly pulling it up towards me. At some point, you can actually see our shadows on the ice. Yeah, there's our shadows on the ice. So light, this is in Lake Simcoe, light's actually penetrating many tens of meters down deep into Lake Simcoe. It's light is warming up the water and driving currents. It's also leading to algal blooms. So a big paper that was first really pointed this out was by Stephanie Hampton, who's a prominent limnologist in the United States. And she pointed out that the amount of algae you have during summer actually isn't that much more than you have during winter. So winter is this kind of overlooked time of year where there really is a lot of stuff going on. And even though it's -20 on this above the ice, below the water, it's about 2 degrees. The fish are just fine. There's lots of oxygen usually. There's light, there's plankton, there's a lot of stuff going on. And so, at the end of the video, let me come out of the water, and there's a picture of me with a beard. Yeah. Yeah, it was really, actually really cold that day, like about -20, but under the, just a few feet down, it's a very stable environment for biology. So we have a bit of an idea about what the general temperature stratification looks like in lakes in Ontario. In summer, we have a warm water over cold water. So on this axis, I've got depth versus time, and the colors representing temperature. As we go into autumn, the surface layer cools down and gets deeper. There's a period of mixing. Water continues to get cooler. Then it inversely re-stratifies. We've got 4-degree water at the bottom, 0-degree water at the top. Ice forms, mixing in spring, and then gets deeper again. So that's been pretty well understood, I'd say, for, I don't know, 50 years, 100 years. What the new area is, what's the chemistry under the water column doing? What's the oxygen doing? What's the plankton doing? So this sort of sketch I made is what we think is happening in Lake Simcoe. So during summer, the bottom of the lake's getting very low on oxygen, so low that it actually doesn't provide any habitat for most of the important fish species. At the end of summer, about half the lake has less than 3 milligrams per liter. It's lethal for most fish. There's a big period for mixing where it renews. Over winter, some work we've been doing suggests that actually under the ice we get a massive increase of oxygen. There's so much algal blooming, it's creating oxygen. Then you've got this cap of ice that prevents it degassing. So the most oxygen you ever gets at the top of the ice under winter. At the bottom of the lake can get a little bit low in oxygen. Then this mixes in springtime and starts up again. So what's important is actually what happens in winter carries over into the summer. It sets the initial conditions for how much oxygen is in the water before the lake stratifies and depletes. So this 6 months in winter, it's not a time you can ignore. It actually is really important for setting the initial conditions for summer. So as an example, this is some data we collected last year. What we do in lakes typically is we don't go out all winter. We put a weight on the bottom of the lake. We have a line that goes up. The buoy stops 5 meters below the surface. And every meter we have little sensors on a line going from 5 meters to 45 meters in the lake. And they measure temperature, oxygen, some of them measure light, conductivity. And we can make these graphs where we have temperature versus time and oxygen versus time. So there's 4 periods here: the fall overturn, winter stratification, spring mixing, summer stratification. So during fall overturn, they're all the same. The water column's homogeneous. All the loggers measure the same temperature. They cool down until we hit 4 degrees, and then we get the stratification. The bottom layer is all near 4 degrees. The top layer gets very close to 0 degrees, and then ice forms at the surface. And in springtime, there's this overturn, the whole water column is homogeneous until we get to about 10 degrees. Then the bottom water is cooler and the top water is warmer. So you see summer stratification is really long. Spring and fall overturn is pretty brief. And then there's that several months of winter stratification that hadn't been previously studied. And so we knew what happened in summer. In summer, we start off with lots of oxygen. It decreases during the year. And then the bottom layers keep on decreasing until they basically hit 0 oxygen at the end of summer. And the longer summer gets, the worse that's likely to be. You can see these surface layers have more oxygen in them. But what's interesting is what happens in winter. In winter, the bottom layers lose oxygen. But this is the exciting bit. Near the top, it actually goes up. So it's supersaturated in oxygen. There's a massive oxygen bloom under the algal bloom under the ice that creates oxygen. And so you've actually had an increase in oxygen. You started winter with about 11 milligrams per liter of oxygen, you end with 13 milligrams per liter of oxygen. We actually oxygenated the water over winter. So if you want to know what the conditions at the end of summer are like, you need to know what happens at the beginning of summer, which really means you need to know what happens in winter. It's probably one of the best documented lakes in the world, but that's the project we've been doing for the last few years with the Ministry of the Environment to try and understand the importance of winter processes. Okay, for time, too long. Okay, I'll go through this next bit quickly. So why do we care about oxygen? We care about oxygen because fish have very particular habitat preferences. So the thing that's most known about fish is if you're going out on a lake and you're trying to decide where do I put my lure, you often want to know where's the layer in the lake where the fish you're hunting for is. So if you're looking for cisco or trout, you want to go to this cold water, it should be deep in the lake. If you're looking for pumpkin seed or bass, you want the warm water, so you'll be at the top of the lake. So if you've got stratification in the lake, there'll also be stratification of where the fish are. In addition to that, fish have very specific oxygen preferences. So some fish like brook brown trout, they want to have cold water that's also got very high in oxygen. And there's some fish like northern pike that just don't care and they can cope with low oxygen events. So with the fish temperature and oxygen preferences, they're very structured where they will go in the lake. So for example, walleye are a warm water fish. They do well in warm water that has lots of oxygen. Cisco like cold oxygenated water, and so they can't go in lakes that have low oxygen habitat. And so with my former PhD student, Brian Flood, and Jill Brooks, who's a PhD student at Carleton, we've been looking at where are the fish in lakes and how do they actually respond to the temperature stratification. So the new technology that's kind of exciting about this is it's a company in Halifax called Vemco is making these tags. So you catch a fish, you make a small incision, you put a tag inside it, and then for 3 or 4 years, you can listen to where the fish is. We put these receivers out so we can monitor the fish all year round. We can know where it is, and then we can ask, why is it using certain habitat? And how do we, once we understand the habitat it uses, how can we improve that habitat to have favorable outcomes for the fish? And so in the Great Lakes, in general, there's at least 1,000 of these receivers, and there's probably at least 10,000 or 20,000 fish that have been tagged. So there's a really exciting story about what the fish are doing. So in a thinly stratified lake, if there wasn't anything happening, you just think cold fish should be here, warm fish should be here. That'd be the end of the story. The thing that's interesting, lakes are actually quite dynamic. When the wind blows, you get these layers forming that move up and down. And Lake Ontario, that could be 20 or 30 meters, they're moving up and down over the course of the day. So it's not a trivial change, it's quite a dramatic change. And I'll see if I can demonstrate that with a hairdryer. If we blow on the lake, we've perturbed that interface, and this will oscillate up and down. It led to some mixing, but it's moving up and down as well. So this was a bit of a mystery, what was happening in Hamilton Harbour. Hamilton Harbour, this is depth versus time, and the colors are representing temperature. In the summer, it's got this warm temperature on the top, cold temperature at the bottom. There's no oxygen on the bottom, lots of oxygen at the top. So you'd expect to see walleye should always be above 7 meters in the lake. That was the prediction. If you actually go and look at the ends of the harbour, though, they're really dynamic. You see the temperature's going up and down all of the time, and it's just look at that in isolation, it doesn't make any sense. What you could imagine is the wind's blowing, and it's tipping the thermocline up and down. So what does that do for the fish habitat? So particularly if you go to the ends of the harbour and you measure the oxygen, during the course of a day, it can go from having lots of oxygen to no oxygen, lots of oxygen to no oxygen. It's incredibly dynamic, and that's because this wind's blowing back and forth. So if we superimpose on that where the fish are, the little dots where we're tracking the fish are actually avoiding these low oxygen layers. They're moving up and down to escape them. And so if there was just random what they were doing, you'd expect there wouldn't be any correlation between where the fish are and where the oxygen layer is, and we'd see a sort of homogeneous distribution. But if they were correlated, the fish should always be above that layer of low oxygen. And so this is the most exciting graph I think I've ever made, because on one layer we've got biology, on the other layer we've got physics, and there's almost a one-to-one correlation where the fish are choosing the habitat that's above this oxycline. So the fish are Sometimes they're 6 meters deep, sometimes they're 1 meter deep, but they're always adjusting to that position where the fish is. So there's a strong, really strong correlation between what the physics of the lake's doing and where the fish are choosing to have their behavior. So I think that's really a bit of the, I've given you a bit of a description in this talk about what the physics of lakes are. We understand that pretty well. We're starting to understand what the oxygen and plankton dynamics are. But the real frontier, I think, for research in the Great Lakes is how do the fish respond? And so there's this really exciting project that's been up for about 5 or 6 years now called GLATOS, the Great Lakes Acoustic Telemetry Observation System. It's an interdisciplinary project between university researchers, provincial agencies, and then state agencies on the US side. And so there's about 150 researchers. Each of these dots is one of those receivers. Lake Ontario is completely filled in now. And in a few years, the entire Lake Michigan, Lake Superior will have receivers in them. And so we will have year-round records of what the fish are doing. Many of these we're also instrumenting with oxygen and temperature. So we'll get a sort of a network of what the fish are doing. And we'll be able to understand how actions on algal blooms and oxygen are actually affecting fish management. So a lot of the research I'm most excited about right now is really working with fisheries people to understand the observations of what habitat fish are using and trying to make sense of the spatial data. So, you know, if you're imagining a little, would a fish choose to swim around at the bottom? Driven by the temperature and oxygen habitat that's available. Okay, so that was my quick introduction to how physics and biology interact and how it might be related to, might shift with changes in the climate. If you've got any questions, I'd love to take them. Yes.
Yeah. First thing I want to say is with the water demonstration, you're clearly having way too much fun. I'm concerned about agro blooms, and you know, we're lucky and privileged that we're not around Lake Erie, and I'm assuming agro blooms are a concern.
Yeah.
Right? So, and agriculture, thank you. I'm talking about agro blooms, and agriculture has a significant influence on that, correct?
Yeah, that's my understanding.
Yes. So is there something that needs to be done, like governmentally or something that needs to happen so that the agriculture has there's more control over what goes into the water?
Yeah, I think this is moving away from science more into politics. And so this is, you know, this is a big economic question. So it costs money to change how you do farming practices. And, you know, most farmers, they work really hard. My granddad was a farmer. It's a hard job. And no one likes being told how to do their job better, and especially by city people telling the country people how to do their job. So it's a political challenge to do this in a way that is a win-win situation for everyone. You know, where when I've talked to representatives through the water pathways of farming units, you know, a lot of them think they're doing a really good job, and they think, you know, you know, we're trying our best to conserve nutrients. So it's not it's easy to point the finger and say you should do this, but politically that's really challenging. So particularly in Ohio, I gather that's very challenging. You know, the state politics of like farmers vote for one political group, city people might vote for another. Farmers don't want to see their incomes being affected, but a lot of this comes down to, you know, how do you grow corn? A lot of it's going for ethanol to replace petrol. The pork consumption. I think most of the pork in the United States is actually grown in Ohio. So it's a difficult mix of economics and politics. I think George will think the science is fairly well understood.
Are there other things that can be done about agro blooms? Like, are there other steps that can be taken?
Well, ideally, you'd capture all the. So when we use the bathroom here, this does not go into Lake Ontario. It goes through a tertiary treatment plant, and we take out a lot of the nutrients. What happens to those nutrients? A lot of those nutrients actually end up to fertilize farm crops to grow corn, and then it gets washed off the cornfields into the lake. So we're saying it's the farmers are doing it, but it's actually, you know, it is us that's originally the source of a lot of these nutrients. So it's a difficult question. Oh, I meant to show these two books by Dan Egan, The Life, Death and Life of the Great Lakes, and his new book, The Devil's Element, are really good. This entire book is about phosphorus and nutrients. And so if you wanted to delve deeper into the severe challenges we have, and on one hand, this has led to the green revolution and like abundant food for the planet. The cost has been these nutrients go into the lakes and the rivers and they pollute our water supply. So Dan Egan, he's a scientific journalist from Wisconsin or Minnesota. It's a really good book, came out 2 years ago. Has anyone read either of these? Okay, well, I thoroughly recommend them. They're good. They're very well, it's a good piece of journalism and they're very well written. Yeah, this one's more about general water quality issues, invasive species, water level. This one's specifically about nutrients. Yes?
Over the years, with climate change and water temperatures changing, is there studies that show that fish have adapted? That fish have adapted? Do they adapt at all to their environment as it gradually changes?
So I'm not a fish biologist, but my understanding is it's more you have winners and losers. So there's some fish that do better in a warm environment and some fish that do worse in a warm environment. So it more tends to be, like if you're looking at Ontario as a whole province, the certain zones of where the fish are doing well might move north and south. So fish that are used to find at 45 degrees might now be doing better at 50 degrees. And so there's a sort of a shift in the ecosystem. So it's happening very quickly. Fish don't evolve that quickly, but it's more that the composition of the population is shifting. So, we're seeing species in Ontario now. So in Australia, for Tasmania, it used to be really cold, and now sometimes you're seeing like tropical fish, like flying fish are coming down on the East Australia Current and are resident in the waters. So there's all around the world in the ocean and the lakes, there's a sort of northward shift of populations. And so that's happening. The shift is happening a lot faster than any evolution.
We have a question online. Great talk, thanks so much. Cisco execs once called people with smartphones citizen sensors, since their aggregate behaviors could signal important events. Is there a future in wildlife tracking where fish act as proxy biosensors, signaling important physical and chemical changes in lakes?
I'd say it's pretty hard to track where fish are. I mean, this new technology is really exciting, but it's not a live tracking tool. There has been a big shift with the, so GLaTOS, all this website I'm showing right now, it's part of the Great Lakes Observation System. There is a big shift, I'd say, with getting live readings. So the sort of measurements I've been making like Simcoe, I don't get that data until I recover it back in May. There's a shift now, a lot of sensors are live. So I can go to a lot of websites now and I can see what's happening in Lake Erie right now. So the research project we're doing Lake Erie, it's got live telemetry. A lot of the systems around rivers around Toronto, you can go and see what the water level is, the speed. This instrumentation of a lot of lakes is going to become more and more so. So there's a big stream of data. What you do with the data, though, is the real question. So actually getting information about the state of the lakes isn't really a challenge anymore. It's more the intellectual activity to interpret it and actually do something useful with it. Like, yeah. So I think that's more the shift. Rather than using fish themselves as biosensors, it's the sensors that are in your smartphone are getting so cheap and so easy to use that we can have an Internet of Things where we have live sensing of a lot of water systems. The real question is, what do you actually do with that data from a management perspective? Any other questions?
There's another.
Yeah.
Another one online. Is there research being done to measure the effect of warm water being dumped into the Great Lakes by nuclear and fossil gas fuel generating stations? These stations dump large amounts of water, which they have used for cooling purposes at 5 to 8 degrees warmer than the water they withdraw from the lakes.
Yeah, so I think they're regulated. They can't do heating more than 5 to 8 degrees. So that's, and if you go down in winter to the beach at Pickering, you can see the steam coming off the water in Lake Ontario where that warm water goes into the lake. So when they were commissioning a lot of these nuclear reactors, they did do studies about what the effect of this would be on the lake. The thing to remember, this is like an inland sea. It's so vast, it doesn't have a large scale effect on the lake. I have heard of it in rivers. If you have a reactor on a river, it can have a big, river's very small, it can have a big effect on the heating downstream. So the Ontario Power Generation is pretty secretive, I'd say, about what they do, but the conversations I have had with them about do fish get damaged by being taken into the plant? Actually, the fish actually really like the warm water. In winter, you actually see more fish offshore from Pickering than anywhere else on the North Shore of Lake Ontario. They're actually they're seeking out this warm water habitat. So it's often not really clear that the biological response is bad. If this was a very small river, I think it'd have quite a different answer. But for a big lake like Lake Ontario, surprisingly, in winter, the habitat isn't bad. In summer, it's not so clear, but you can't see it. It's not a very strong signal. The deep water cooling that N-Wave does downtown in Toronto, that has a similar effect. You're pulling in cold water and releasing warm water back to the lake. But because it's such a large lake, it's really negligible, the effect it has. So other places are doing this in the US. Cornell University on Ithaca is doing this. I think Zurich and Geneva and Switzerland are using lake cooling as a thermal regulatory source to get clean energy in summer and winter. So it does have an effect, but it's I think by keeping it to that small level, it's not, surprisingly, but it's not negative for fish.
Perhaps leads into the next question a little bit, which is research similar to yours also being done in the ocean?
Oh yeah, yeah. All the good ideas I have are things people developed in the ocean 30 years ago. So I'd say, the real advances in a lot of the fluid dynamics really come from the ocean. And partly there's a lot more money in the ocean. A lot of it comes from naval research. And so if you want to understand internal wave dynamics, a lot of that was funded by the American Office of Naval Research to understand how their submarines would work in the ocean. A lot of the technology driving communication and sensors is driven by the ocean. The lake community is pretty small, so it's really advances that are happening in oceanography that are flowing over into the lake research. So all the sensors I use, the lake market's pretty small. They sell most of them to oceanographic researchers. So yeah, so let's say I follow what they do in the ocean, not the other way around. Although interestingly, the word limnology was actually invented before the word oceanography. There was a Swiss-French researcher out of Lausanne on Lake Geneva, and he wrote a book that I've got upstairs. It's in French, and you look through it and you go, he had all the good ideas 120 years ago. And he defined this word limnology, monomictic, meromictic, epilimnion. And that was about 5 or 10 years before the word oceanography was defined. So limnology is a little bit older as a science, but oceanography is, orders of magnitude bigger in terms of a discipline. Any other questions? So the main thing about this demo I wanted you to take away is that it's still there. It's been for an hour. This red layer, clear layer, they've persisted. They're very stable. If I don't perturb it, nothing really happens. So that's not how you generally think about water. You generally think about water as being mixing, like a river. But in a lake, I can heat it and cool it at the surface, the wind can blow on it, but these density layers are persistent and they're not obvious. When you're going around on a boat on the surface of the lake, you don't know what's happening 50 meters down or 100 meters down, but it's really important for driving a lot of the chemistry and the biology of the lake. So if you don't remember anything else in a few weeks, just that demonstration's the main thing to take away. Thanks.
Thank you very much for your presence. Just want to let you know that our next seminar is scheduled for November the 7th, and Professor Cosima Porteos will present on climate change, NEMO's new threat. Okay, I think we have the room until noon. So feel free to enjoy our coffee, refreshments. Okay, thank you very much.
Make sure you get a parking exit pass if you haven't already gone.